I would like to talk to you during the next 15 minutes about the impact of the European In Vitro Diagnostic Regulation 2017-746 on laboratory testing for rare diseases. This is obviously not a very sexy topic this late in the evening, but it is a very important topic, in fact, for rare disease testing. The European IVD regulation is a European regulation, which means that it becomes binding automatically for all EU member states on the date it enters into force. This will be in May 2022 for the IVD regulation. The aim of the regulation is to improve the quality of in vitro diagnostic tests, just like the MDR, the Medical Devices Regulation, which enters into force in May 2021, aims to improve the quality of medical devices. One of the drivers, in fact, for the MDR and also then the IVDR were scandals such as the PIP scandal in France with the breast implants. The major principle of the IVD regulation is that it is important to have a stricter regulation, and that as much assays as possible should in fact be CE IVD registered, and that these assays should fulfill a series of criteria for validation, including, and this is new, clinical performance. So these assays should have a proven intended use. The impact of the IVDR cannot be underestimated. While under the old IVD directive, only around 7% of the CE-IVD assays had to be certified by an external organism, this will increase to more than 80% under the current IVDR. As certification has only started, it is already now clear that not all assays, not all manufacturers will be able to register all their assays by May 2022. The IVDR, however, also, and that's in the small print, restrict the use of so-called in-house lab-developed tests after May 2022. While many laboratories throughout Europe mainly use CE-IVD assays for their routine chemistry and hematology, for example, lab-developed tests play an essential role in, for example, testing for rare diseases. The Federal Agency for Medicines and Healthcare Products is the Belgian competent authority for the implementation of the IVDR. That means the FMHP has to oversee the implementation in Belgium. There is some leverage for member states, but in fact, this is a European regulatory framework. We have been working for a number of years already, since the law is from 2017, together with the FAMHP and the geneticists, the Commission of Clinical Biology, the Commission of Pathology, and BELAC and Sciensano in a working group chaired by Nathalie de Vos from LHUB to prepare the implementation of the IVDR. And we also have established a number of subgroups, including a subgroup for molecular biology and genetics, and one for chromatography and biochemical testing. However, we have not been able to make a lot of progress, as we are, in fact, just like FAMHP, waiting for some additional European guidance. And the COVID-19 pandemic has obviously not improved the ability of Europe to prepare the implementation of the IVDR. In fact, the MDR has been delayed for 1 year, so it should have entered into force in May 2020, which was delayed to May 2021. There's, however, it's currently not clear that the IVDR would be delayed. So the date is currently still May 2022. What is now, in fact, a lab-developed test? There is, in fact, no definition as such contained in the IVDR, but based on the regulation, one can conclude that an LDT is an in vitro diagnostic test which is manufactured and used within a single organization, a single health institution, and not put on the market. Currently, these kind of assays are typically widely used for rare disease testings, where in fact there are typically only a few commercial assays available. Infectious disease testing, where pathogens can change regularly. The respiratory panel and SARS-CoV-2 would be the typical example here. And genomics, a fast evolving scientific field. It is important to understand that the IVDR will give preferential treatment to CE IVD registered assays. These assays are typically obviously commercial assays because registration and maintaining a registration costs money. So everybody can register his own in-house assay as a CE IVD if he pays to these external organizations, but obviously that is something typically which companies will do with a commercial interest. The assays which are registered as such have to fulfill a series of requirements, including clinical performance and post-marketing surveillance. And LDTs can only be used in such cases where there is a specific patient need for which there is no equivalent CE-IVD assay on the market. In fact, the requirements to be able to use this exemption are clearly defined within the regulation. And these include that the device cannot be transferred to another legal entity. You cannot make it in one institution and share it with another institution. You have to be either ISO accredited as an institution, not for the test, or where applicable, which is the case in Belgium, there should be national provisions regarding accreditation, which means that in Belgium, the Praktijkrichtlijn Guide Pratique covers this requirement. You need to document that there is this unmet patient need. You have to be able to provide information to the competent authority. You have to draw a declaration which can be made publicly available. This is still all relatively feasible. However, there is also the requirement that specifically for Class D, these are typically devices for which you would never use a CE-IVD unless there was none available, for example, testing for blood components for transfusion. There are a whole series of requirements regarding manufacturing facility, manufacturing process, which have to be prepared. Member states have some maneuvering room, but can also apply the same provisions to the other devices. And Belgium, in fact, has this intention, which could mean a significant amount of extra work for laboratories. Member states can also, and that's not to be taken lightly, restrict the manufacture and use of specific assays. So if the FAMHP comes, they can just say that you have to stop using your assay. And final paragraph, which is important, the exemption does not apply for devices used on an industrial scale. What this exactly means is not defined, but that's a form of uncertainty which could cause trouble in the near future when a commercial company would say, yeah, but this is this industrial scale anyway. You claim there is an unmet patient need, or the assay might not be as good, but you manufacture it on industrial scale, you have to register it. We did a little case study in our own hospital to see what the impact would be. How many LDTs do we use? Our hospital is a large university hospital. We perform approximately 17 million laboratory tests per year, which obviously are a lot of liver function tests, hematology tests, etc. But if we then looked, we compiled a list of all our laboratory tests at our Department of Laboratory Medicine, excluding the microbiology cultures because it's a bit difficult, obviously. You start with a single hemoculture flask, but you can have multiple cultures starting from the single flask. We included histopathology and constitutional genetics because these are other laboratories in our institution. Tests were classified in 4 groups: a core lab with routine chemistry and hematology, immunology, including autoimmunity testing, the special chemistry lab, which we're focusing on here now, which includes rare disease testing, toxicology, special therapeutic drug monitoring, inborn errors of metabolism, and molecular microbiology. Each test was then classified as CE-IVD. You use it according to the manufacturer's intended use, and according to his instructions, you modify it. You use a different washing buffer, different volumes, etc. It's research use only, which in fact means it cannot be used for diagnostics. This will not be possible after May 2022, or it is really such a lab-developed test. We considered any modification as such as a different washing buffer or different pipetting volumes as a modification. It's unclear what the impact of this kind of modification will be on the CE-IVD status. But a typical example of how manufacturers treat this would be Roche. Roche on its automated immunoassays has blocked the possibility to do increase and decrease run standard for parameters for which they haven't validated this. So they consider even the use of different pipetting volumes than they have validated as a modification, which is not allowed under the IVDR. Not to be forgotten is that all matrix tests which are not validated by the manufacturer are also off-label use. That means if you use an amylase test to test a fluid from an abdominal drain, that's definitely not a CE-IVD test. And you might also be required to register this assay with the FAMHP and make a validation dossier, etc., etc. When we look at the total volume of tests, which is more than 12 million for our department, we have almost 98% of CE-IVD results. But if we look at the level of the laboratory tests, the picture is completely different. Less than half of the tests we offer are in fact CE-IVD. What do we see? We see for the routine platform, there are a lot of modified testing. This includes some coagulation testing, all this kind of fluids that we test, etc., some flow cytometry, and also definitely for the special chemistry laboratory, tons of lab developed tests and only a couple of CE-IVD assays. If we quickly look at some of the typical reasons to use lab developed tests, these are typically the main reasons there is no CE-IVD assay. So in fact, many of these assays will not enter in, will not cause any problems under the rule that there cannot be an equivalent device on the market, but will have to fulfill all the requirements, including all the paperwork. There are a number of modified assays, off-label use, etc. If we quickly look at the special chemistry assay, in fact, only 11% or 12% of the assays were CE-IVD, and these accounted for less than 30% or 1/3 of the results. The main reason was obviously the lack of CE-IVD methods for rare diseases, typically in toxicology. Moreover, there are also a couple of assays, typically for endocrinology, where there are some CE-IVD methods, but the analytical requirements, for example, you want to measure estradiol and estrone in postmenopausal women receiving aromatase inhibitors. That's not possible using commercial assays, for example. What is now the potential impact of this regulation on laboratory testing for rare diseases? The aim of my talk is mainly to raise awareness, but if you have a look, and that's the idea, you need to also look at your own tests, and then we also need to work together to also see if there are things we have to discuss with the government. A first scenario would be that obviously there are multiple CE-IVD methods on the market for this test, and these meet your analytical requirements. Typical example would be, I want to test glucose in plasma. There are lots of tests which are good, and I choose one of these methods. This is the standard scenario for these 97% of tests we perform with CE-IVD tests, obviously. However, for rare diseases, this is exceptional. The problem is the whole regulation was written from the viewpoint that there are CE-IVD solutions for almost every patient requirement. The next possible scenario is that typically there is only one CE-IVD method on the market which meets your analytical requirements. And there are typically two problems you encounter here. There is typically the risk of inadequate supply. And think about the example of Roche being the first company to have a PCR test for SARS-CoV-2 detection, but already within 2 weeks, they had supply problems. In fact, the IVDR poses no obligations on suppliers to guarantee supply. So it means that when the CE-IVD company, then the supplier of the CE-IVD assay can't sell you the kit, you're in trouble and you can use an LDT. But if they call you within 2 weeks and they say we have a new supply, you need to stop using your lab developed test. There is no obligation on suppliers to make sure that there is adequate supply. And this is definitely a problem. I have to admit that I already encountered this regularly for rare diseases. Another possible risk, which I haven't encountered yet, as we could use LDTs in the past, but if you would be the first one to register and the only one to have a CE-IVD assay for a rare disease, you also have the risk of an exorbitant price. This is not an example which is so bizarre. The typical example for people in the field of inborn errors of metabolism would be the chenodeoxycholic acid, which is used for the treatment of a number of peroxisomal disorders. I took this from the internet today. There was the case in the Netherlands, for example, where the UMC, so there's a fusion of AMC and the VU in Amsterdam, the two university hospitals, where they make it themselves, this compound, because Leadiant, the company, bought all the companies in Europe making the drug, which was sold for less than 30 cents for a capsule, and then just increased the price to 140 euros. There is no problem whatsoever in the European regulation for rare diseases. There's only a problem for the healthcare insurers. So this is not some exorbitant example. This has been done in the past, and there is no reason to assume that Europe will do anything against this. The next possible scenario will be a relatively not infrequent scenario, is the situation where there is some CE-IVD device on the market with an intended use which somewhat covers your use, but not meeting your analytical requirements. Because, for example, say you have a neonatal screening kit which can detect acylcarnitins. I want to do acylcarnitins, but I want to chromatographically separate methylmalonyl carnitin, succinyl carnitin, for example, for people in the field, in plasma, not in blood spots. Yeah, That's a different. That's different. But what situation are we here? So there is no device on the market which meets our requirements. There is some analytical device on the market. In fact, it is your requirement as a laboratory to continuously prove that there is no alternative. You have to continually look in Europe if there are not any CE-IVD assays available. It's still unclear what would happen if you would miss the fact that such an assay had become available. Moreover, there's also the continuous risk that the regulator or a court could not accept your argument and suddenly would be forced to use the CE-IVD assay and stop the use of your LDT. Moreover, there's also the risk that you will have to switch to an equivalent the day this equivalent becomes available. This means that the assays that you're running today in your normal spectrometry, on your mass spectrometer, you will continue to run them today, but will you buy a new machine? Will you buy a new platform for lab-developed PCR testing, which could detect SARS-CoV-2, or will you switch to closed commercial platforms which do not offer this flexibility? So in the medium term, this is definitely a risk for the European citizens. Finally, you have the situation which is the most common one. There is no CE-IVD method on the market. However, there is still a risk here because the additional regulatory requirements and costs can result in the fact that the number of tests that are available will decrease. This is also not so bizarre. I'm thinking in Brussels about the MPS 3 San Filippo, where the University Hospital of the UZ Brussels stopped offering testing for MPS 3 after they were sued and they lost a court case. They said, okay, leave it as it is. We don't offer it anymore. There are no laboratories offering MPS 3 testing for all the different San Filippo variants. So it's just not available anymore. So there is a risk that the number of tests that will be offered will decrease as regulatory requirements and costs will increase. And there's also here again the risk, will laboratories, when they will have to buy this new mass spectrometer, for example, for this assay, will they buy this machine for which they need at least 8 or 10 years to just pay off the equipment without making any profits? Will they buy such a machine not knowing whether they will be able to perform this assay for all these years? I would like to conclude after this brief overview that it's clear that the IVDR will restrict the use of in-house laboratory tests after May 2022, and that a case study in our hospital found that although most assays, more than 97%, were generated with the CE-IVD method, less than 50% of the tests we offer were CE-IVD. Moreover, there was in fact, in total, but certainly for rare diseases, no equivalent device available on the market now. I'm not even talking about the devices which might not receive approval under the new IVDR. For more than 70% of these lab-developed tests we are offering. There is in fact currently also an urgent need for European guidance for the implementation of the new IVDR for lab-developed tests. This is clear. We only have a little bit over 1 year to go, but there are still a lot of uncertainties. My main message for tonight is that the deadline is approaching, and it is important that we all try to prepare ourselves. It's clear that we don't know everything yet, how we will have to implement it, but 2022 is not far away. So I would advise all of you to prepare yourself. Thank you for your attention.